Clicked for IB Biology. Now we're going to take a look at what is called the secondary structure, the second level of organization of proteins in the structural hierarchy of protein structures. In the description there's a PDF that you can download to annotate your own notes, and you can take the quiz afterwards to make sure that you've understood the content of what we are studying. So just to frame the context, remember that we've already looked at the primary structure, which is the order of the amino acids in the polypeptide chain as dictated by the gene. These chains don't exist as straight chains in the cell. What happens is that as they form on the ribosome, they immediately will form up into what are called alpha and beta forms, alpha helix and the beta plated sheet. And that's the structures that we're going to look at now in this video, and then we'll follow that up with further levels on tertiary and quaternary in the next video. In the determination of the secondary structures of proteins, this was attributable to the great Linus Pauling, thought of as the father of biochemistry in the 20th century. Pauling worked out of Caltech in Pasadena, and like the university, was well known for his rigor in his scientific work, and indeed the university's reputation was very much established by Linus Pauling. So some of you may become so proficient that you get to go and work there. So anyway, Pauling in 1951, believe it or not, determined that the primary structure would fold up into one of these two shapes here. So the chain of amino acids will fold either into an alpha helix, which is a right-handed helix, or it may fold into what is called the beta pleated sheet. Now the reason for this folding is that in the polypeptide chain we have groups like this sticking out, the oxygen double bonded to the carbon, and there are also hydrogens sticking out off the hydrogen. So what happens is that the hydrogens on a nitrogen are attracted towards the double bond oxygen on the carbons, and this reoccurs at various stages along the polypeptide chain, causing the whole thing to fold up into this helix. The diameter of this helix is approximately 1 nanometer, and the pitch where it repeats its pattern as it turns around the spirals is about 0.4 nanometers as it repeats itself in the structure. That is the repeating of the coils. Within each of the turns of the helix, within the pitch, there are approximately 3.6 amino acids. These details were all determined by Linus Pauling using surprisingly low-tech methods of a protractor, a ruler, a piece of paper, and some sticky tape. Pauling then quickly realized that there was an alternative structure to the secondary, which was that it might actually fold up into this zigzag structure that you see in this diagram here. So this is the beta pleated sheet. And again, the hydrogen bonds occurring are here to here, and then here to here, here to here. And in each case, although one hydrogen bond is not particularly strong, many hydrogen bonds give the alpha helix and the beta pleated sheet stability in their structure and shape. The beta pleated sheet is noted for its high tensile properties, making it a very, very strong structure. And indeed, it is found in spider web, in the silk of spiders. So it is that strong. Of course, they are single polypeptide chains. So the single length of the polypeptide folds up into the helical structure. And in this case, it forms up into the zigzag structure. But of course, at some point, that zigzag structure has to do a turn, like so. And we know that where this, so that would be out here, in fact. And in those positions there, we know that the amino acids there are glycine and proline. And that can actually become important in later aspects of our study. Primary structures fold into secondary structures. The secondary structures have two forms, the alpha helix and the beta plated shape, both determined by Linus Pauling, and a core part of a student's understanding of the structure of proteins. The links below now take us to the tertiary quaternary level of the hierarchical structure of proteins. And we have a button here for you to subscribe to this channel, and indeed a link to the playlist on proteins.